And so I got the recording warning. And so you'll get a little bit of a preliminary taste, but this has been a part of my research for since 2017, so maybe about 6 years. And so I'll mostly be talking about some of the preparatory work that put me in a position to be interested in plant microbe interactions. So to start with, does anyone know what legume this is? This is really obscure. I'm just curious. I'll give you a hint. It's neotropical. Nope.
No.
Okay, this is Schwartzia. It's an Amazonian legume with some of those really crazy nodules on the right-hand side. So nitrogen fixation is a globally important symbiosis. I don't think I have to argue too strongly about that because I can just cite the fact that it is central to our food supply, especially protein. And in natural ecosystems, nitrogen fixing symbiosis is very important to nitrogen cycling. It's where nitrogen mostly typically comes from, although there are a few other sources. It is a complex relationship, and it's all based on the fact that nitrogen is very plentiful on Earth, particularly at something like 70% of the atmosphere, but that is a triple bonded gas molecule. And so it's super stable. Most life cannot at all access most of that nitrogen. There are a few groups of bacteria only that are able to do this through an enzyme called nitrogenase, where they're able to overcome that crazy strong bond and make things like nitrites and nitrates, which are the bioavailable forms of nitrogen. This is complex from a lot of different perspectives. Just like the slide shows, you can go all the way from genes and cells to systematics in the environment. I'm a systematist still, and so I'm going to give you more of a biodiversity-oriented perspective. Actually, even if we just narrow it down to the diversity of symbiosis, things get really complex. But I'm going to first start by narrowing things down to talking about nodules. So not all nitrogen-fixing symbiotic plants are involved with nodules, but the most important ones are in terrestrial ecosystems. And so this includes legumes, of course, the nodulating crops and many other plants. So this nodule is a specific housing organ that hosts the bacteria, mediates the transfer of photosynthate to the bacterium. That's what's in it for the bacterium. And then in return, the bacterium gives nitrogen to the plant in an available form. Now, what is a nodule? This is kind of a complex question. So on the left-hand side, that's what nodules often look like in legumes. This is little doodads that are on the end of roots, typically small spheres or hot dog-looking things. And if you pull a clover or a pea plant in your garden, you'll probably see something just like this. That is actually the nodule of Medicago, which is the typical model organism for the legumes. Now, actually, really quick, I'll point out one more thing about that. You can see that the big hot dog shaped nodules on the left are bright pink or red. That is leg hemoglobin. I'll mention it in a minute, but it is responsible for excluding oxygen, which is hostile to the nitrogenase activity. And so this needs to be an anaerobic process, no oxygen. You might also have heard of leg hemoglobin as a key flavor compound in Impossible Burgers. So that's what legume nodules look like, but there's way more other stuff going on. I'm going to try to turn on my laser pointer. And now we can look at this elder nodule. That's Ulmus and Betulaceae. And that looks like this brain-like kind of orangish mass. They're really striking. Or we can look at Ceanothus, maybe on the East Coast. This is less familiar, but I think in pine savannas or whatever you call it when it's further north, you might see these. California lilac, you may know it as an ornamental out west. Bifurcating, strange, white-looking roots. The normal roots don't look like this. So they're still quite specialized. Well, it turns out that nodules are all over the place. They do have one thing in common. We're quite certain that they are lateral roots. So these are root organs that are modified as housing structures. That even includes the ones that occur on the stems. And so you don't have to be part of the root system to be a root nodule, as it turns out. Now, I was just stressing the diversity, and so it might not surprise you to learn that everything else you may know about nodules is not universal. I think in this audience, there may be some people that have taught this kind of content or learned it in courses, maybe even seen it in talks. You may have heard about symbiosomes, these famous organelles that are in the cytoplasm of the cell that host the bacteria, so the bacteria actually in the cells. Not all nodules do this. The signaling molecules that I teach in my intro plant bio course, not all nodules do that. That leg hemoglobin as well. None of those are universal. Not even necessarily universal to economically important nodulators. So they're very diverse. Now, the next question we can ask is, who are the nodulators? Well, there are, as we understand it now, 10 families in total that can do this. This is according to the most recent phylogenetic classification that I'll discuss in just a minute. The legumes, of course, are the top of the list. If you knew one nodulating plant, it was probably a legume. That includes pea plants, these kind of puffball mimosa type plants, and many others. These, as well as this very obscure Pacific Island lineage called Parasponia in a different family, they both interact with rhizobia, another word that maybe some people in the audience have heard before. Rhizobia are nitrogen-fixing bacteria. They're actually a general term for a number of different genera and families that are not that closely related to each other, but they are proteobacteria, so one of the bacterial phyla. The classical ones, including the Rhizobium, which is a smaller genus than it used to be, are alpha proteobacteria. In more recent times, things, especially like these puffball plants, mimosa, may be nodulated by beta proteobacteria, which are quite different. Well, that only accounts for 2 families. The other 8 families have a completely different bacterial partner. That is currently called Frankia, a genus in the Actinobacteria. That one, if you've heard before, if you've heard of actinorhizal plants that have these relationships, I'll be very impressed. But you may have heard it because actinobacteria are very important soil organisms. So those families include members of the rose family, Dryas. It's actually the earliest branch in the rose family is all the nodulators. Elaeagnus, this whole family only contains nodulators. You may know them as ornamentals or as terrible invasives, probably slightly south of your region. California lilac ceanothus I've already discussed. So now I can answer, where are the nodulators? So this is just the backbone APG4 classification. So this is botanists' most recent consensus classification for the flowering plants. And we'll only fuss about a little slice of them, if I can get it going. That is now called the nitrogen-fixing clade. This comprises 4 orders: the legume order Fabales, the rose order Rosales, the oak order Fagales, and then the melon and curcubit order Cucurbitales. This is what we now call the nitrogen-fixing clade. Traditionally, before molecular data and molecular systematics, we had no idea, or they had no idea, because I was barely born yet, but it wasn't known yet that these were plants that were closely related. That was the breaking result of this seminal paper, 1995, in PNAS by Soltis et al., chloroplast gene sequence data, et cetera, et cetera. So that was the key finding, that all the nodulators are fairly closely related. So now I'll give you just a little bit more detail. This is a photograph that I prepared a long time ago representing the nitrogen-fixing clade. With our new tree, things might be just a little bit different. But this is a small slice of the flowering plants, but the flowering plants are so diverse that there's still a lot of species. So 10% means over 30,000 species. Lots of familiar plants, some I won't have time to mention today, but you may recognize them around the edge of that tree figure. The branches in that circle tree are actually colored by the bacterial partner. So the rhizobia that I mentioned are red. The actinobacteria that I mentioned, or sorry, the rhizobia are blue. The actinobacteria that I mentioned are red at the bottom of the figure. And the non-nodulators are in black. So these are things that don't make nodules. I think you can see by eye that somewhere around 1/2 to 2/3 of this group has nodules. They have nitrogen fixing symbiosis. It's actually really mostly just one giant subclade of the legumes, maybe a second one here that I'll talk about in a minute. That accounts for almost all the nodulators on Earth because the legumes are mostly nodulating in their omega family of almost 20,000 species. The non-legumes down here have very sporadic symbiosis. So you can see lots of red branches, or very, very small amounts of red branches spaced out by lots of black branches. So very common colored symbiotic species up here, very rare colored symbiotic species down here. That's a pretty big dichotomy in diversity, I would say. And so that's one of the things I've been interested in for a long time. So Soltis et al. 1995, they not only found this result of all of the nodulators being closely related, but they had a hypothesis that this represented a predisposition to nodulation. Their hypothesis was not that the ancestor of that clade could engage in symbiosis and nodulate. Their hypothesis was that some sort of a change that made it easier to gain symbiosis happened in the ancestor. They felt that that was probably some sort of a genetic change. Now that we know a little bit more about how genes underlie traits, it could be a gene, it's probably regulatory elements and similar things involved in expression. So anyways, the predisposition hypothesis hung around for a long time is more or less what people thought. But about 2 decades later, right at the close of that second decade, this pair of papers came out that tried to quantify this a little bit because the Soltis et al. Argument was sort of eyeballing a tree, although it was a major accomplishment at the time. Particularly, Werner et al. Used a model-based approach to directly test the predisposition hypothesis, which they strongly supported. So that's great, but not all was well. So there were some advanced modeling techniques that were used in these papers. The phylogeny that underlies it, less so. And so this is, just to pick on one of the papers, this is one of the supplemental figures straight out of Lee et al. 2015, which was the second one I cited in the previous slide. So this is a tree based on one chloroplast gene, MATK, and so a fairly limited amount of data, at least by modern standards. But that's fine. It seems like there's a number of support values in the backbone of that tree that are okay. I see some 80s, 90s, and 100s. Let me draw your attention to the highlighted genus names. Those are, at the time we assembled this early proposal, the legume genera in this group that were known to nodulate. They're in yellow, the non-nodulators in black. If you really try to squint hard and look for a pattern, it's not really there. It looks completely random. Actually, it looks scrambled. And so I was suspicious of this at the time. And I, along with other people, especially those in the legume community, felt that even though these modeling techniques might be important, the underlying phylogenetic data were not good enough to actually tell us where on the tree nodulators arose because there were too many uncertainties. Almost at the same time, actually, I guess the papers hit a couple years later, this pair of papers came out in Science and PNAS. This is Griesman et al. 2018, Van Velzen et al. 2018. This was the next development in this business about the predisposition hypothesis. These were comparative genomic studies that basically just sequenced a bunch of nodulators and non-nodulators in the nitrogen fixing clade, and they just queried a bunch of genes that we know are essential for symbiosis. People have done experiments. You knock the gene out, they don't do nodules. Okay, so it turns it off. So it is essential. And most genes are not interesting, but 2 or 3, depending on which paper you look at, were, particularly the ones that were shared with NEAN and RPG. I'll show you those on the next slide. So the nature of the data in these studies was actually fairly simple. The figure on the right shows just a presence-absence matrix. So it's like checking a bunch of boxes. The columns are genes and the rows are species. There is a tree on the side here, and this marks the nitrogen fixing clade. We won't worry too much about what the specific lineages are for now. The remarkable thing they found was a pattern of gene presence absence, best shown by NEAN, that I have highlighted with these big red letters. NEAN is a signaling molecule, so it's really at the start of symbiosis. So it's involved in sensing signaling. And in the outgroups here, these are things that are not in the nitrogen fixing clade. NEAN is always around. It's doing something else. It actually has to do with ectomycorrhizae, so working with fungi. It's been known for about 10 years that a lot of the molecular machinery for nitrogen fixing came from those earlier fungal interactions, especially the signaling. Something different is going on with NEAN when you shift to within the nitrogen fixing clade. All the nodulators, pretty much, they're actually marked in the blue on the other side, have NEAN in a full copy, so they're marked in black. The ones that do not nodulate tend to either have white, so there's no copy of the gene, or gray, so there's a copy, but it's broken somehow. That's what we would call a pseudogene. Like it has some sort of a mutation that makes it fragmentary and not able to code. So the pattern is not perfect, and it is less so for RPG, but this is basically a correlation argument that is saying that this is a very early gain of function in these genes, probably in the ancestor of the nitrogen fixing clade. Now, these two papers were very confident in their interpretation. They felt that this was a home run evidence that the ancestor of this clade, if you were to rip it out of the ground, would have nodules on it. Our immediate reaction, because I was doing a postdoc with the Soltices, so maybe a slight conflict of interest, but we were curious about the degree to which this evidence is different from the predisposition hypothesis. So some people felt the case was closed. We didn't think so, and we still don't think so. So that's where the NitFix project comes in. This is something that's been going on since late 2017. It is a large-scale genomic effort to get a couple hundred genes from almost a majority of the nitrogen-fixing clade. I think the real number that we've achieved, at least in our first paper, is about 12,500. That would still make it one of the best comparative systems in plants for doing lots of things, such as investigating the origin of nitrogen fixation symbiosis. Here's the main personnel. I'm mainly going to talk about the work of 2 postdocs, so just to highlight them. Heather Cates on the top right there was the main postdoc at the University of Florida. Now she's in a data science position. And Carol Siniscalco was my postdoc at Mississippi State. Now she's in a tenure track data science position at the Mississippi State Library. So you can tell this was a big data intensive effort. I am reporting, first of all, on the results from this preprint, which we're hoping will come out in Nature Communications this year. This was our flagship paper on investigating the origin of nodulation. So to start with, I'm going to spare you the details of fussing through a very large tree because those can be pretty dry talks. But I do need to give you a broad strokes update of what we found. And actually, the story is quite simple. So this was a very large scale sequencing effort. Actually, Smithsonian was involved, mostly through Ting Chuang-Yi's lab at Kunming Institute of Botany, and a number of other major herbaria. And through that partnership, we were able to sequence many, many species that had never been sequenced before. Some had been sequenced for only one gene. So we really didn't know where these species would go on the tree. That was really exciting. And so we were expecting surprises. I think I'd argue that the surprises were of a fairly easy to describe type. I'll remind you of the supplemental figure that I was picking on earlier. Random distribution of nitrogen fixing symbiosis. Those yellows were all over the figure, no pattern. This is what we see now, kind of the same area, a little bit of a different zoom of the cisalpinioids, some of those that have those fuzzy puffball flowers and legumes. The black are the non-nodulators. Now they're falling out in these very nice branches, very clean on the tree. And that was exciting because it turns back the clock on the origin of symbiosis. Probably fewer evolutionary events, probably earlier in time. Definitely post-Cretaceous, right? So not in the age of the dinosaurs, but not too long after. Now, the other thing that I think will be a really important outcome of our study, that's the red branches here. Those represent a sister clade, so most close relatives. One has nodules, one doesn't. These are ripe to develop new experimental systems, and they've never been known before because we didn't know where those species go on the tree. So it's really important information. Unfortunately, our key legume organism, our lab mouse of the legume world, is Medicago, which is probably in the most boring place imaginable if you care about symbiosis research, because it is deep, 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 deep in those papilionoid legumes with pea flowers that I mentioned before. So that's enough about the tree. Now I'm going to tell you a little bit about the model. I'm just going to keep this fairly simple, but I think it's important to understand the story just a little bit. Depending on the kind of stuff you do, I know we have a couple molecular systematists in the audience. Sometimes you come across these things called hidden Markov models, especially if you work on gene families. If that's not familiar, basically, this is a class of models we use to try to estimate an unobserved parameter. So there is something about nature that we can't directly measure, and we're trying to estimate it through proxies, essentially. These have actually been used for a number of years on investigating predisposition hypotheses like the one I've been talking about. This Evolution 2012 paper, Marazzi et al. In the bottom right corner, is the first application in systematics. This actually was on legumes, but it was on nectaries, not on nodules. And there are very cool models, but that's all I'll say for now, because if you get me talking about models, we could be here for a while. Again, for any molecular systematists that might be in the audience, if you know HISI, a diversification method, it's also closely related. So we're trying to basically estimate variation in how fast something happens in evolutionary time. And we do that by estimating a trait in which species have it. So some species have a trait that cause high evolutionary rate. Some species have a trait that cause low evolutionary rate. And so that's the hidden part of the model. And the trait that has high evolutionary rate but does not have nodulation, that would be the definition of a predisposition. I'm going to throw this result at you, and I'm going to ask you not to worry about it too much because I'm going to simplify it in a minute. But I'll start by saying we tested a bunch of different hypotheses for how many times nodules originated. I had mentioned when I talked about genomic studies that those two authors were very confident in single origins, right? So they thought that symbiosis happened once in Earth's history, nitrogen-fixing symbiosis in a nodule, and everything that doesn't have it in that clade was one of thousands of losses. That model in this data set, we definitively reject even when we mess with a bunch of our assumptions. And so we're very confident in that result. It's also a mixed story. We have gains and losses. So this is a complex story about the history of these plants. Now I'm going to simplify this figure and show you what we think is going on. This is a rate matrix. It looks complex, but those arrows just represent how many events happen per millions of years. So a thick arrow means something very frequent or rapid, and a very narrow arrow means something didn't happen very much. Now that concerned the categories here. We've got 4 of them. So I'm going to walk you through those, and I'm going to do it in time order from the beginning. So the very earliest ancestor of the nitrogen-fixing clade was in what we call a precursor state. So this means that our model strongly favored the predisposition hypothesis. So we are consistent with that 1995 Soltis paper after all these years. And that's the beginning. Our model actually showed something a little more complex than the earlier paper. We actually identified a second precursor state. And so every single plant that gained a nodule and could do symbiosis actually had 2 steps to gain. That's where the name of the paper came from, 2-step process. And we're trying to represent that by these gears here. It's sort of a cartoon, and they represent potentially different levels of genes, genetic architecture, regulatory elements that may underlie these changes in evolutionary lability that we reconstructed. And so the first layer is the orange one, the second layer is this red one, and then when the gears nicely tooth together, that's how we're representing the concept of the phenotype of symbiosis being gained, not just the genes. There's one other category here. This non-precursor is an important one because these are plants that don't have nodules, but they're not precursors. They actually have no path to gaining nodulation, right? And so according to this model, you could run the evolutionary clock and they would never gain nodules. The reason the model thinks this is there are large parts of the tree, especially, those rose relatives, oak relatives, and melon relatives, where for, almost 100 million years, symbiosis has never changed state. They've never had symbiosis. And so this is the state that represents what's going on with those plants. To make it a little more concrete for you, I'll show you that in relation to overall diversity. Those tiny figures around, or pictures around the side, those are trying to show some of the familiar plants. If you know your legumes, there's so many of them, they occupy 2/3 of the figure. I'm pointing to the root of this evolutionary tree. That's where we get this precursor state. So that is exactly what Soltis et al. 1995 said. There was something that happened in the ancestor. Then, right here, this blue, we get a big gain of nodules that led to a huge diversity. That's about 1/3 of this huge clade, so at least 10,000 species. These are the papilionoid legumes, the legumes that have flowers that are like pea plants. So that's where a lot of the diversity comes from. And most of the rest of the diversity is the mimosoids that contain these puffball flowers and some other types. Here's where all the other origins are, all over the tree, very isolated little branches on the total tree of life, kind of like that story I told at the beginning. Again, it's a story of many gains and losses. The losses are here, but they're very small and recent, and so they're hard to see. Just to kind of summarize things in a table format, we have 10 families that have nodules. 9 of them are not legumes. And of those, there's only one origin in each, either the whole family or a really early branch in the family, with the exception of Rhamnaceae, where we were able to clearly resolve exactly 2 origins. No losses. Within the legumes, things get a little more interesting. Maybe some of you may know that the legumes have been shaken up recently. So now we recognize 6 subfamilies within the legumes. We are quite confident. We did a big revision of our knowledge of symbiotic states for this paper. We're quite confident that only 2 of them can nodulate or have any members that can nodulate. So of those, 3 origins in Sessile pineoides that contains mimosa and relatives, 3 origins in Papilionoides that contains peas and relatives, and so equal numbers of gains in each of those. So they're not that different. So the non-legume nodulators, there are very few of them. They're very tiny branches on the tree. If we look where they are on the globe, they're also not that widely distributed. You might say they weren't very successful. This is also true, actually, of most of the legume origins, except for 2, the mimosoids and the papilionoids. So we know that nodulators are all over Earth. You probably have them in your yard. They are members of these 2. So something very special happened. So not all nodulators are the same. Symbiosis is a big deal, but just because you have symbiosis doesn't mean you're able to reap all the benefits, perhaps. Now, I want to slightly refocus for the kind of second half or third of this talk, because I become very interested in this concept of loss. This isn't something that has received a lot of exploration or discussion historically, mostly to the extent that people have looked at this, and unfortunately, a lot of legume biogeographers don't. But to the extent that they do, it's presence absence. It's whether you have a symbiosis or not. The ones that don't are not the same. There are some that have never had symbiosis that I talked about a while ago. There are some that only lost it very recently. I think that those species are a special window on the understanding why a plant engages in symbiosis in nature. We know a little bit, of course, about why they do it in agricultural systems, right? It's all about nitrogen. What's going on in nature where they're often found in nitrogen-rich ecosystems, or they're not found in ecosystems that are hurting for nitrogen in particular? So what environments are promoting loss in particular? What environments are promoting more symbiosis in species and more different types of symbiosis, like I introduced at the beginning of the talk? And I think loss is a key part of this. So I'll give you a whirlwind tour of some of the papers that we're working on recently on this topic. This was the first that came out recently in Global Ecology and Evolution, Global Ecology and Biogeography. This was last year, and this uses the neon sites that were just mentioned. So the 45 terrestrial neon sites, they are highlighted across this map, and the number of nodulators is the little red slice on those pie charts. And the size is the species diversity for whatever neon site that was. And the key result of this paper was trying to figure out what promoted 2 things. One is what kinds of environments result in rich communities of symbiotic plants. So which environments favor communities having many species that can nodulate? And then second, we didn't want to just count species. We wanted to estimate something called phylogenetic diversity. So we wanted to understand how distinct the lineages were, the branches on the tree, that are involved in symbiosis in these communities, right? Keeping the species number the same, are they very different from one another in evolutionary terms or not? And the reason goes back to my introduction to this talk. Those lineages are an excellent proxy for diversity of the symbiosis, particularly who the bacterial partners are, because that's a lot of what we're capturing. And the key kind of breaking result of this paper, and there's been a couple of others kind of around the same time, is that the key environmental parameters shaping how many nodulators you have in a community, it's about precipitation, which is not the traditional explanation for this, which is more based on nitrogen, limitation. This was a super interesting result for us because we actually wrote the grant that this is related to, trying to look for things like soil predictors of where we find nodulators on Earth. And it turns out some soil predictors are important. Nitrogen is actually one of the least important. So it does a very bad job of telling us where we find these plants on Earth. Now, of course, the symbiosis is about acquiring nitrogen. So nitrogen is part of the story, but nitrogen availability need not be. Partly linking with physiological literature, and we do talk about plant physiology in this paper. One of the things we think is going on is that nitrogen is very closely related to water stress. And so this is why precipitation is favoring nodulation. So dry, arid areas are favoring more symbiotic species. Plants put most of their nitrogen in photosynthesis. Actually, almost a majority of it goes into rubisco, which is the enzyme that actually takes carbon dioxide out of the atmosphere. To take carbon dioxide out of the atmosphere, you have to open your pores up and let water out. So water costs to plants are also very closely related to photosynthesis. Something that's been known for a very long time is that nodulating plants have higher leaf nitrogen by far than non-nodulating plants from the same sites. It's quite a stark difference. And so what we actually think is probably going on is legumes are simply, and other plants that nodulate, are simply making different nitrogen investments. So their needs for nitrogen are not the same as their neighbors. And so that, I think, explains a lot of this discrepancy. So this is the neon sites. I'll come back to that in a minute. We're also doing global scale work. And so we've been doing a bunch of species distribution modeling. This is looking at the mimosoid legumes. And this color palette is showing you where loss of symbiosis is distributed, because in mimosoids, we're quite confident that the ones that have symbiosis are all recent losses. If you know your plant geography, those green and blue areas are where the losses are, and they have something in common, right? They're all tropical forest. Most of those are wet tropical forest and lowland, so rainforests. The highest richness of loss of symbiosis on Earth is in lowland tropical forest in West Africa, sub-Saharan West Africa, by a large margin. And we built a model to try to explain how environment relates to this. This is a little like that rate matrix I showed you before. A big arrow means that variable is super important. And precipitation, both aridity and raw precipitation, dominate the model here. You can just see that by the arrows. Soil does play a role, temperature to some extent. We did test nitrogen. Nitrogen is actually not in the model. Soil pH is related to nutrient availability, potentially. So that makes sense. So again, another story about precipitation. Those non-nodulators are specifically associated with very wet habitats. And so what we think is going on is the lack of drought stress is disfavoring symbiosis. When I first worked on this, I found that to be kind of a profound idea. Like, why would you lose symbiosis? Why would you lose a mutualism, as we would call it when both partners benefit? When I took Intro to Ecology, we learned about this by little checks and X's where we marked who wins and who loses. Competition, both lose. Parasitism, one wins, one loses. In mutualism, both are supposed to win, but that's conditional on the services each one is providing. And this is a very expensive relationship. About a quarter of the photosynthate, of the products of photosynthesis the plant makes, are eaten by the bacteria. That's a pretty big number. That's like on the order of your metabolic cost as a human being for having a brain. Now, this figure is showing that same map of how many species that lost nodulation, and the top is showing you how many species of these mimosoid legumes there are. If you look at the middle and the top, I think you can just eyeball, there's no relationship, okay? The richness of Africa in differences in symbiosis is not reflected in species number. Africa is not that rich for this group compared to some other areas. The bottom, however, this is again phylogenetic diversity. We're counting in relative terms how much evolutionary history is localized in the species in each community. Tropical West Africa is the hotspot on Earth for mimosaurid lineage diversity, and it lines up very nicely with loss of symbiosis. For a variety of reasons, we think that this reflects the environment filtering different groups of plants that lost symbiosis that we now know are separate losses. And so the environment is actively selecting against symbiosis. Again, these very wet areas where there may be no drought stress. The last part of this project is we're trying to compare this across different branches of the tree of life. So at the beginning of my talk, I mentioned rhizobial plants. That's what I've been showing you on the right. And I'll show you an actinorhizal plant, Phagales, on the left. This includes a lot of your familiar forest trees and 3 independent origins of nodulation. First of all, you can tell that the distribution is super different. This is something that's been known for about 2 years, that the bacterial partner seems to be related to niche specialization. The rhizobial plants are most prevalent in the landscape in subtropical to tropical areas. And when you get to very cold areas, the non-legume symbionts tend to be a little more rich in comparison. So that's what's being shown in Phagales on the left. In the southern hemisphere, we see a precipitation relationship just like we were expecting. These really arid South Africa, Southeast, Southwest Australia, these areas have a high proportion of nodulators. This is actually the same color scale, by the way. In the north, it's different. So precipitation is still the most important, but the relationship sign changes. So the wetter areas have more nodulators. We're not exactly sure what's going on here. This seems to be very specific to certain families that are not legumes. However, for a lot of reasons, there can be a lot of parallels between dry adapted and aquatic adapted plants. They actually have some similar physiological pressures, one of which is carbon dioxide limitation. And so we're exploring some other factors that might explain what is probably a local radiation that is very specialized to potentially some nutrient poor, very wet habitats. Last thing I'll show here is this is more of a phylogenetic look at comparing aridity and temperature, specifically looking at losses of symbiosis versus those plants that never had them. So loss is green, never having it is white. These are violin plots, and so they're just kind of showing the spread of the raw data. The plants that lost symbiosis are present in both fairly arid areas and some of the wettest areas in our data set. So there's a little bit of bimodality that we'll be exploring in there, possibly a somewhat complex story. And they're very disposed to tropical temperatures. And so for the most part, we see a relationship that's close to that concept of a lowland tropical forest and losing symbiosis. The last thing is this project that was mentioned in the very kind introduction. This project is 2 months old as of now, but we're really excited about it. And it kind of comes from a common reaction when I used to give this talk, which is I talk for almost an hour about the distribution and diversity of the plants. And what does a really smart student in the audience ask me? They ask me, well, what's going on with the microbes, right? Because I didn't actually tell you anything about bacteria. I told you about plants and I wove a little bit of a story about the bacteria, but we really don't know exactly what they're doing in natural ecosystems. It also derives from a perception that we had a gap where a lot of people do studies on plant microbe interactions. Everyone and their brother does it these days. And a lot of people do studies of nitrogen fixing bacteria. But at the scale we were looking at, the really global scale, we didn't find anything we could use. And so a lot of people disagree on things like whether how specific the interaction is between plants and microbes. Do the plants care what nitrogen fixing bacterium is in them? Is there any coevolution between them? Or are they adapting to each other? That's something that people get really passionate about, and everyone has a different answer. And it could be that differing geographic and taxonomic scope is the problem, and we need something broad in order to be able to answer those questions. So we're looking at going all the way from the local soil environment to the hyperlocal soil environment right on the roots. It's what we call the rhizosphere, all the way into the nodules. We want to understand at the neon field sites which nitrogen fixing bacteria are available for symbiosis and which ultimately get recruited for symbiosis. We want to do this across lots of species and especially across lots of environments. So this figure on the left, this is some of our preliminary data. This is just showing going from the soil to that rhizosphere that I mentioned, the soil that's right against the roots, to the root to an actual nodule. This is a stacked bar plot showing you what bacterial species are most prevalent. All the colors contain nitrogen fixers. Okay, so you can see this nice progression into nodules, which for the most part tend to have like a 99% superinfection by one strain of nitrogen fixing bacterium. Not always though, but most of the time. We want to document that process. We want to look at diversity. In this pilot study, we've been looking at some localized areas and different plants. We need the neon data to get the climatic variation, and that's the core of our work that we're trying to do. We now know that the plants are responding to precipitation. That really seems to be what controls it. I want to know, is that what's controlling the bacterium? And is it because the bacteria have similar pressures, or is it due to symbiosis itself? The last thing I want to ask is, are these microbes co-evolving with their plants? Are they selecting on each other, living together over millions of years because we know this symbiosis is so ancient? So I'm working with Claudia Solis-Lemus, who is a statistical phylogeneticist at University of Wisconsin-Madison. And with her, I'll be applying some of the latest and greatest in these fancy interaction network techniques. And so what we're going to try to do is quantify how specific the plants and the microbes are, how much based on phylogenetic data we think they're co-evolving. And we want to do that going from wet environments to dry environments, from temperate environments to tropical environments, because we think that probably the stressors of climate would be the key input that would make this symbiosis really important, and might also make it important for human beings in the future when we're facing a more challenging landscape and where nitrogen fixing crops could be very important. I would like to close by thanking, first of all, my lab at Mississippi State. I'm in my 5th year, and so I've had 4 years of great times with these people. One of the undergrads is shown on the right there on this project. And my long-term collaborators, Rob Graunik and Doug and Pam Soldis at the University of Florida. Carol Siniskalki was my postdoc on this project. I already mentioned she's got a job now, so she's out of the lab, but we're still working together. And the Department of Energy, and especially the National Science Foundation, for repeated funding of this work. And I should have remembered to put the Smithsonian, I hate myself for not doing that, but a very large network of herbarium partners that made this work possible. I didn't make a big deal about it, but everything I've shown you today, specimen data are deeply integrated in the background. And so without the generosity of curators and of investment in collections, nothing I would have shown you is possible. I would love to handle any questions that you might have.
Wonderful. Thank you, Ryan. Thank you. Questions?
Hi, this is Gene Rosenberg here. I noticed on on one of your maps, and this is of the U.S. actually, it was a proportion of nodulators. There was a super hot spot in Southwest Georgia, and yet, I mean, they're just I'm sitting here in Miami. They're just the neighbors, and boy, it doesn't seem very arid at all. I was wondering what's going on there.
Well, so the thing is, I guess that you're probably focusing on, I think that's the Jones Center is the name of that specific site. Um, so precipitation isn't the only input here. And so, if we just built a model that only contained precipitation, it would be a lot worse than the model that we have. But the it's really that result wasn't just about raw precipitation. It's specifically about aridity. And the metric that we actually use in that paper, it's not just the amount of the precipitation that the plants get, but also their potential evapotranspiration. So a little bit of a closer window on the exact drought stress they might be experiencing. That could be high in the Western United States as well. And so I think it would be really great to look at that more longitudinally rather than latitudinally, I think is what you're keying in on. And we might discover that there are some other things that are really important. I wonder if edaphic ecology, other than nitrogen, may be one of the things, especially soil acidity. Um, Joseph, I think you've been raising your hand for a while.
All right, thank you. Great talk. So the fact that symbiosis is lost in the tropics among the legumes implies there must be some cost to the plant from the symbiosis. What is that cost? Is the bacteria creating some damage to the plant? So it's partly pathogenic, or is it some metabolic cost, or what do you think?
So the photosynthetic cost is probably the main thing. It's about a quarter of the photosynthate the bacteria eat up. So that's big. But it's funny that you mention it because actually parasitism can be one of the costs. So that's one of the things we want to investigate in the new project because one of the reasons why we think that coevolution should be happening between bacteria and plants is that bacteria are always able to cheat and take more than what they're supposed to get, or just provide nothing. And so you start to get a net negative interaction. And we know that at least legumes have some physiological mechanisms for potentially ejecting these or aborting the symbiosis, and that may vary between species. So I think that the main thing is the metabolic and specifically photosynthetic cost, but it is important to consider that continuum between positive and negative interactions because there probably is an arms race going on between the two because it's too tempting to cheat when so much is at stake.
Got it. If there are no other hands raised, I have a second question. Was the flip side with greater loss among the non-legumes in the northern latitudes, and could that be explained by pH? I mean, I think bogs and sphagnum, and so another variable that was in your model.
Yeah, that's one of the things I'm thinking about. pH was in the model, but to my recollection, I still think precipitation slightly outweighed it, although they probably have some interaction. So I think that edaphic ecology is part of the story. I still think there's something going on with aquatic environments. There actually are some aquatic legumes as well that might fit into this pattern that are not cold northern things. In your region, maybe things like Sesbania, senna, and so forth that often live in like really low quality kind of river scour environments. And so there may be an ecological specialization involved. So, we build a global model. Maybe these plants are doing something slightly different, even though we may have been successful describing the overall pattern. So maybe the summation I have is stay tuned, because I'm hoping we can figure out some of those things.
Got it. Thanks.
Yes, Mary.
So I have a question to follow up with your observation about what are areas having problems with CO2 availability. And is this related to stomatal opening? And so is there when there's a lot of moisture, is there a lower stomatal opening?
That is not quite the argument. Now, I'll confess that is me going out on a limb. So take this with a grain of salt. But another complex trait that has a very similar history is CAM photosynthesis, right? Where they can change their photosynthetic mechanism to acquire carbon differently.
Mm-hmm.
CAM photosynthesis is known from arid growing plants. It is also known from several aquatic plants. This has been worked out, and it seems to be that this is related mostly to limited diffusion of carbon dioxide in aquatic systems. And so it's really hard for plants to get as much carbon dioxide as they need because they're underwater. And so they need a carbon concentrating mechanism. None of these nodulators are completely aquatic. They generally just have their feet in the water. That's usually where the nodules are. Even that has exceptions. There are stem nodulators, and they are all semi-aquatic legumes. I don't know what the story is, but I think
very cool.
Yeah, I think there's a story to be told, and it's at least my pet hypothesis that I hope carbon dioxide has something to do with it. Oh, really quick, carbon dioxide actually is in experimental systems related to symbiosis. If you put something like soybean in a climatron and you accelerate CO2, you make it really high, they are able to fix nitrogen much faster. So at least in experimental systems, there's something going on there.
Very cool. Thank you. Ryan gave us a lot to think about.
I scared you off. All right. All right. Well, if there are no more questions, thank you very much, everyone. Thank you for
I
the
can ask just at least one more question. Following up with there's just you've taken such a huge chunk to look at. I mean, it's amazing what you're achieving, and how broad this is. But do you think phosphorus might be important? And when you're thinking about I know it's crazy to have to think of so many things, but in the edaphic factors that might really be playing a role that you haven't had a chance to look into.
We have not actually had a chance to look into phosphorus, just like you're saying. For background for other people, even in your garden, nitrogen tends to be the thing that is really short. It's usually what's limiting plant growth. If it's not nitrogen, it's often phosphorus, although usually it's kind of weird soils on Earth that are short on phosphorus, but there are areas of Earth that are pretty big that are short on phosphorus. I think especially arid areas. Absolutely. I think there's a role to play. Once again, in experimental systems, you can set up something where nitrogen fixing symbiosis responds to phosphorus, right? Because it responds to photosynthesis, plant growth. It's all closely related. I don't have a story to tell that specifically about phosphorus, but I will say that I'm hoping that we can include it in our models, because I think there's some good global phosphorus data sets that we could potentially leverage to do that. And try to link in with some of those agro-ecosystem experiments where they actually do look at phosphorus pretty frequently.
Great, thanks you. All right.
I can ask one more question. Just because
Sure.
I'm always thinking about polyploidy these days, and it seems like every plant has done something with its genome. Is there any correlation? Is having an extra genome worth of genes helpful in being predisposed to forming nodules, or it's not related?
Well, it's funny you ask because I had a slide on this subject and I hit delete yesterday. Sorry about that. I may have misjudged my audience slightly. It was actually because it's kind of a side project. So there was a Nature paper, must have been about 13 or 15 years ago, Cannon et al. And they showed that there was a ancestral polyploidy event, they thought somewhere before the papilionoids, right? So those P legumes. And I'm not sure they had the sampling to know exactly where it is. That gets tricky because you need a lot of genomes for that. And there's a lot of papilionoids. But there is a polyploidy story. And they speculated at the time, they felt that it was related to the gain of rhizobial symbiosis. And this has actually been something that has been slowly, I think people cite it a lot. They still think so. We're actually working on a paper kind of closely related to this. Don't tell anyone. It's actually not that big of a deal. That Van Velzen paper, 2018, that I had cited, that was on Peristonia. One of the reasons they felt that there was only one origin of symbiosis was that they thought they caught a gene duplication in the act, in the ancestor, and one of the copies became the nodulating copy, and the other one still does mycorrhizal stuff. It turns out we have reason to believe that polyploidy created the extra copy in the legumes, but in the non-legumes that have two copies, it wasn't polyploidy, it was a tandem repeat. So they just repeated a piece of chromosome. And so that duplication is not the same thing, which is very important to our multiple origin story, but it doesn't necessarily downplay the role of polyploidy, which may very well be related to especially the legumes. And duplication is important because most of this machinery was stolen, borrowed, whatever, from ectomycorrhizae. And what's a very easy way to make extra copies? Well, we'll just double everything, right? So we don't know exactly what polyploidy is doing, but it's definitely something important.
Thank you.
Great question.
Right?
Yes.
I think I have how much do we know about the mechanism in the Frankia system? It's
yeah, go ahead. Sorry.
yeah, so much more seem to be emphasized on the rhizobium, and so the Frankia system seem to have a lot of more times of evolution, and they just seem to not have driving like diversified as much.
Yeah, that's right. So those Frankia plants, or actinorhizal plants, as we might call them, they're ancient. They're all over the place. They're not that successful, depending on what you mean by that word. They're doing something very different. People are working on the genetics behind it and kind of the genome phenome thing. One of the tricky things is I think a lot of them just kind of suck as experimental systems. They're perennial, they're all woody. That's not the best thing if you're trying to do like, crossing plants, inbred lines, developmental studies, which is extremely important for nodules because you always want to catch the infection process. Some of them don't even make nodules until they're quite old. Some of them have weedy strategies. So Casuarina, she oak. Some people are working on that. Well, Parasponia is rhizobial, but it also has this problem, but it's kind of weedy. A couple labs are working on that. And
Well, how much is on like Rosacea?
not much that I know. I think there's some experimentalists on Dryas. There was a paper that came out this year in Nature Plants where they did some developmental transcriptomics of Dryas. I imagine it's a tricky experimental subject because it likes glacial moraine and like super cold habitats. Once again, not my favorite system if I were an experimentalist. I'm based in the deep south and it's very hard to keep things like this alive. So I wouldn't want to be doing it. So like I said, people are working on it. One problem that I have with a lot of the studies that are trying to look at single origins of symbiosis, the other thing about the Frankie assistant is they're so different from one another. For instance, Phagales, the ones that I showed a map of, they're missing LCOs. They are missing the first bacterial signaling step that initiates the process. That is a big difference. They have a different signaling compound. We don't know what it is. And you know, they don't have leg hemoglobin. The bacteria have their own way of excluding oxygen. They probably don't fix as much nitrogen as the legumes do, just because of, you know, Frankia grows slower, a number of other reasons. And they have these weird habit things, like a lot of them are perennials. They're kind of slow growing. They're temperate. A lot of them live in very strange soils like serpentine soils. And so there's a lot to explore and probably not enough labs that are working on it.
Right. But I was thinking about in the sense of not successful or successful, but ecologically, the Frankia system seems to be extremely successful. Those plants, they may not have enough phylogenetic diversity, but they occupy the ecological diversity is really high.
Yeah, that's right.
Northern hemisphere is completely dominated by them.
Yeah, that's true. That's fair to say. The diversity is not so high, but if you're in the right ecosystem, they can be extremely prevalent. Like, you know, you go in a bog and Murica or Morella is everywhere. In the Tombigbee River, we see a mile of alnus trees right along the coast. So in the right habitat, they are fairly prevalent. I still think some of them might be a little bit more ecologically marginal than, say, the legumes. But they're very diverse. They have a big palette as far as habitats that they can occupy. And so I know it gets really tricky when you start talking about success, because then everyone is like, well, what do you mean by success? Because if you make the definition different, then different plants will be successful. So I definitely grant your point.
Is it known which one is older? Which one of the two symbioses is older evolutionarily?
Well, so by older, so I don't think we consider one to have led to another, but if you just mean like which one came first in time,
that's what I mean.
Yeah,
I don't think. Yeah, no, I was not that one led to the other, but
in ours, we've done some time calibration. We haven't done a lot of really well calibrated fossil calibrations. We do have a little bit of a handle on this. The legumes, I think people are pretty confident that they're somewhere around the KT boundary. I think there's a few studies that have shown this. And the nodulation would have been pretty early in that divergence, especially that 6 subfamily thing. They're this tiny little, it all happened very fast. And the modern subfamilies were around very soon after they originated. The problem with the non-legumes is I mentioned how phylogenetically isolated they are. It's very hard to tell when they nodulated, because what you get this nice monophyletic family of nodulators and this giant stem that goes all the way back. Casuarina is an excellent example of this. Casuarinaceae, the family may date to the Eocene or something, depending on what you think of pollen evidence. Some of those are really hard to date. But whatever you think, that branch goes way back in the Cretaceous. And so, without direct fossil evidence of symbiosis, you really don't know where on that branch it originated. So it's a tricky question, but I would definitely think that both are very ancient, and I would be willing to wager that both might have a late Cretaceous history, possibly latest Cretaceous. So I'm not sure.
Thanks.
All right, so thank you very much, Ryan. We probably could go on for a long time now. You've really got the ball rolling. Thank you so much. It was really terrific. Great talk. Much appreciated.
Thank you for the kind invitation. I was excited to talk about this again.
Okay, everybody. So next up is going to be a holiday party. So